Welcome to the presentation of Cell on the topic of autism spectrum disorder, ASD. We are trying to work on new diagnostic and therapeutic approaches to understand autism, which is so far an ununderstood entity. This is the first of three clips. We talk first about the background and afterwards the diagnostic and treatment related research of our Israeli company, Cell. Since autism was described in 1943, that means over 75 years ago, there is actually no good understanding what is the feature, the nature of autism. And we remain with questions that we are lost, confused, and we don't know exactly what to treat. What is the diagnostic understanding, despite a lot of research in genetics and other aspects and psychology? And therefore, autism is called a spectrum disorder, meaning everything can be included because there is no clear understanding. What we try to do in our research efforts is to understand diagnostically what we are dealing with, and based on that, to define therapeutic targets, and based on these targets, to offer treatment. That there is actually interesting observations on these therapeutic aspects, which put everything in a clear picture together. It goes back at least to 1971 that researchers in the field described immune and autoimmune aspects in autism. And here's just a paper from 1971 where in a child with autism there was also the presence of other autoimmune diseases, including Addison's disease, moniliasis, hyperparathyroid diseases, Addison, and diabetes. And later on, based on the case reports, on many case reports and clinical observations, there's a famous milestone paper by a group by Atla who investigated association of family history of autoimmune diseases in autism, and he found a long list of 9 diseases which are found statistically more often in children with autism. That is a clear clinical indication to understand, to search for autoimmune or immune aspects in autism. Our group started with this paper in 2013 and published in the Journal of Autoimmunity. And with a big group of experts worldwide, we were capable to summarize and to review immunological and autoimmune considerations of autism spectrum disorder. And there you will find a lot of insights of our working hypothesis, which is the basis for our research in the field. And I would like to summarize briefly what are the criteria for autoimmune disease. Vitebsky-Rose defined these criteria, and we summarize them with direct proof and indirect proof, as you will see here in a moment. The hypothesis is that in the maternal body, there are antibodies which might affect the baby, the fetus. For the mother, these immune aspects are not necessarily inducing a disease, not pathogenic, but there are the basis, the family history of autoimmune disease, that there is an underlying issue with immunity. During pregnancy, the baby, the fetus, is exposed to these immunological aspects without the capability to fight these immune aspects. So during pregnancy, these maternal antibodies might affect the fetal brain, which is very sensitive during the development, but after birth, postnatally, the child presents clinically with autism, and the mother had these autoantibody called maternal immune activation, and the child exposed to these antibodies during pregnancy is affected with autism afterwards. Or in the young fetal age, in the young fetal period, the child has these exposure to immunological aspects. Going back to Sherlock Holmes, there is a very elegant research performed by major groups in the States, and they checked whether the maternal antibodies injected to a pregnant mouse will affect the offspring. And it was shown that nothing happened when the blood of the mother is injected to the mouse. But if the mouse is pregnant, the offspring of these mice, they will present with autistic feature. And that is a proof of concept that something in the mother who is not sick, has no clinical evidence of any abnormalities, injected to a pregnant mouse, nothing happens to the pregnant mouse, but the offspring exposed to these antibodies during pregnancy will present exactly with these features. So that is a strong proof of evidence. We summarize that with the following graph in our paper in 2013, that there is an evidence, as you see in the right side, the fetal nervous system. We know that there is an impairment of the fetal brain development. The brain is bigger of these children, well known from observations. And there is also a finding, the so-called microglia, which are where there is a swelling in children with autism compared to children uh, not diagnosed with autism, where there is a normal size. Microglia reflect immune cells which fight chronic inflammation. So the working hypothesis is that immunological aspects to the brain of the baby, of the child, or the fetus, they induce changes which are compatible with autistic behavior, as you see on the right side of the slide. Going backwards historically, might be related to the maternal immune system, and through the placenta, these antibodies come through the fetus, affect the fetal developing brain, and we have to search on the left side, both in the mother, but of course in the child and the fetus. And as older the child is, of course, the chances to find something in the mother is reduced, and we are finally treating the child, trying to understand diagnostically the child. So the focus is on the child, but historically it goes back uh, to pregnancy and exposure from the mother. That is the working hypothesis, that intrauterine or transplacental exposure causes the autism. So based on this hypothesis that autism has autoimmune aspects, we search for objective and early laboratory findings, both for the diagnosis and later on for the treatment. The child, we are trying to find from the peripheral blood samples for the laboratory analysis, and our company is strongly involved in this research with very interesting findings so far. We take blood from the child, we try to find antibodies in the system of the child, changes aberrations in the immune system, and we believe that with the so-called AAA autism, we don't call it autism spectrum disorder anymore. Spectrum is everybody, everything is included. We call it a AAA, standing for autoimmune associated antibodies, autoimmune antibodies which are associated with the autism. And we believe with this AAA, we can make major progress, both to understand diagnostically the entity of autism, and of course, most importantly, at the next stage, as we will see in the third clip, to approach these children therapeutically in an intelligent way. Our group had the privilege to publish in Frontiers a year ago, and we have an editorial, and we have a whole volume with our group, and brought there together an interesting team worldwide of experts. And here you see how many people. There are 68 authors, and it has a very good exposure. So to summarize, autism is currently an entity where there is no known cure or any curative approach to treat, because there is no proper basic understanding what we're dealing with. We are trying to find immune markers in order to diagnose these children objectively, and that will be the basis for the autoimmune antibody associated autism. That will be the AAA research result. We are trying to find children at the early stage, because that is the best time to find when there is brain plasticity and the beginning of the process, of course. That will help us both to diagnose and monitoring, and these will be the therapeutic targets that we want to develop later on, therapy which will be a revolution in autism indeed. We focus on the diagnosis and treatment, and we have here a team where we can be approached through our website. We are blessed with a wonderful team. Teamwork makes the dream work. And we hope that with this research, diagnostically and therapeutically, we can make a major progress for the world and all the children in need for better treatment. Thank you very much for your attention. We will continue in the next two clips.